which he gave a couple of months ago. Dr. Schultz is the curator of birds at the Natural History Museum of Los Angeles County. She came to that position in 2018, and it's been a delight working with her since then. She got her undergraduate degree. She's a native to Southern California, and she got her undergraduate degree at University of California, Berkeley, where, among other things, she did the sort of resurvey of Grinnell and others' early surveys and a couple different time periods of the birds of the UC Berkeley campus. And then while she was there, did a resurvey to look at change in bird populations over 3 periods over about a century. She went on to get her master's at San Diego State University, working with Kevin Burns, who's sort of Mr. 9-primaried Ocene and in particular tanagers. And I think it's fair to say that's where Allison really got hooked on tanagers, which pretty much immediately leads to being hooked on color as well. She went on to get her PhD at Harvard under Scott Edwards, and she worked there on a subject that was much closer to her previous webinar on disease and house finches, and really just looking at short-term evolutionary responses to stressors such as disease. So she's very good at crunching numbers. She's very good at understanding of what makes birds colorful and how they make their color and how they use and perceive their color. And that's what she's going to be talking about tonight, which is the eye of the beholder, the evolution of feather color in tanagers. So we're very pleased to have Allison. Thanks for coming tonight.
Thank you, Kimball, for that wonderful introduction. So I'm going to share my screen. So as Kimball mentioned, tonight I'm going to be talking to you about my feather color research. And, just a little bit of intro about me. This may look familiar if you saw it last time. One of my goals being an evolutionary biologist, and in particular an ornithologist, is to understand the diversity of birds and why it got that way and how it got that way, to the best of our abilities. And so there are kind of a couple axes that I like to think my research exists on. One of those axes is to trying to tie genotype or the genetics of a bird to phenotype or general traits. So think of this as like feather color, for example. Oops. And also spanning different time scales, so spanning deep time to recent time. And so I have 3 main topics of my research. One of those is the evolution of plumage coloration. One is urban evolution, which is kind of one of my very recent focuses, and also host pathogen evolution, coevolution. So last time I talked to you all about this main category, host pathogen coevolution. And this time I'm going to stick to plumage color evolution, although there are overlaps. And so what do we think of when you think of the evolution of plumage coloration? Well, you might think of natural selection. So that is one of the big drivers of why birds are the certain colors they are. There are a number of different types of natural selective pressures. So for example, feather colors, particularly colors formed by the pigment melanin, actually make feathers strong. This is an example of a frigate bird that washed up on shore, which was an albino bird. And you can see its feathers were just totally demolished, and probably because it didn't have the melanin molecules which strengthen those feathers. So that's one selective pressure that bird feathers on. Melanin also strengthens feathers against feather bacteria. So, birds are constantly trying to protect their feathers because they're so important for one of their main kind of driving life history traits of many, many birds, which is flight. Birds can also help birds Color can also help birds thermoregulate. So typically you'll see darker birds in more humid habitats and lighter birds in drier habitats. But you might also sometimes see dark colored birds in the desert. For example, this phainopepla, where its feathers can capture that solar radiation and allow that heat to disperse without kind of leaking through to the body cavity. So there are many different types of kind of naturally selective pressures. I've only touched on the surface of a few of those that are helping to drive plumage color. However, that's not the whole story, of course. Here's a peacock, which if you picked up the New York Times today, you can see, you might have seen that article. Apparently they're a big problem here in Los Angeles. But a very famous quote by Charles Darwin was that the sight of a feather in a peacock's tail, whenever I gaze at it, makes me sick. And that's because he couldn't really make peacock feathers fall into this theory of natural selection, because there's no, there's no benefit from a natural selective perspective to have really fancy plumage that's going to actually make you more visible to predators and make it harder for you to get away. And so consequently, the theory of sexual selection was described, where feather color is also used not just to protect a bird against different types of weather or protect against feather wear, but also used in communication with other birds. For example, a male trying to attract females. So bird plumage coloration is really shaped by both these different selective pressures, natural and sexual selection. And there really is a balance between these two things. Now, how to study kind of the impacts of natural and sexual selection? Well, you want to have a group of birds that has a lot of variation in its color. And really, there's no better group of birds than tanagers, at least in my mind, to study the evolution of plumage coloration. Here's a picture of tanagers I laid out. This is actually not at the Natural History Museum in LA, but at the MCZ at Harvard, when we had an event and I decided to make a rainbow of tanagers. Perhaps I will do that again sometime at our museum, but it's really fun. But you can see tanagers, they really do span every part of the rainbow. And one of the things that really makes them great is not only do you have many bright and colorful ones, but you also have many more cryptic species. So, many browns, many black birds, for example, many green birds that are going to blend into the environment. And so just to remind you a little bit about tanagers. So tanagers occupy almost all environments in Central and South America. So not only are they really variable in their plumage, but also where they live. So they occupy many different types of habitat. They occupy many different types of temperature. They eat many different types of food. They have many different bill sizes. They sing many different kinds of song. And all this is to say that it's a really excellent group for studying many, many aspects of bird diversity, including color. And in fact, they make up about 10% of all passerines or all songbirds. So they're one of the largest families of birds that there is. And I put up here Kevin Burns, he was my master's advisor, as Kimball mentioned, and really has used tanagers to great success for studying many different aspects of bird life history, and is my collaborator for much of this plumage coloration research that I will be presenting. So one of the first things that we did when I joined Kevin's lab was to construct what's called the molecular phylogeny of tanagers. This was a part of a larger project to study 9 primary Dacians, where we learned fun things like, for example, our Western tanagers, or all of the parangas, are not actually tanagers, but are cardinals, and so you won't find them in this tree. And that many things that we didn't know were tanagers are in fact tanagers. I could give a whole nother talk on that, so I'm not going to go into it. But I just want to just show you that we actually do know how all of the different tanager species are related to each other. And we categorize them into 15 different subfamilies of tanagers. And these subfamilies, they differed in terms of life history characteristics, kind of what you think of as quote unquote core tanagers, or the Thraupinae. They're the biggest subfamily. These are what contain what are called the mountain tanagers or the Tangara tanagers. And we also have, for example, the Sporophilinae, which contain all of the different Sporophila seed eaters. So things that you wouldn't think were tanagers. And so, okay, so now I've told you a little bit about color and a little bit about tanagers, but what are the specific questions I'm going to address in this talk? Well, those are male or female plumage color changes correlated with the evolution of sexual dichromatism? And I'll explain all these questions in a minute. Do changes in plumage coloration for males or females correlate with the evolution of sexual dichromatism in specific plumage patches, so in specific areas of the body? Do species with more complex plumage have faster rates of speciation? And how do plumage coloration mechanisms evolve differently in males and females? So now for this first question. And so why are we thinking about males and females? Well, again, thinking about this fact that avian plumage coloration is shaped by both natural and sexual selection, And one thing we really want to know is the impact of these two different forces. And what better way to study this than to study birds that share the same genome, so the same genetic history, but might be under slightly different selective pressures, and those are males and females. And so typically, you think of a male as being brightly colored and a female maybe as being more cryptic, because oftentimes in birds, it's the male that's trying to attract the females. And so often they have fancier plumage coloration. However, that's not always the case. There are many species in which males and females are the same color and they might be cryptically colored. And there are many species where males and females are the same color, but they might be colorful. And this is especially true in tanagers. And so we can make use of that to understand the evolution of sexual dichromatism, or differences between the males and the females. And so in kind of what people used to think was that really it was male-driven changes in sexual dichromatism or sexual selection that was causing males to become more colorful. And so that was really what's driving these colorful males. So one might say that perhaps sexual selection is stronger than natural selection. However, there's another possibility, which just depends on what your starting point is, which is that you might have very colorful males and females, and actually natural selection might be so strong as to drive females to become more cryptically colored or more drab colored. And so in this case, you can see how we can pick apart, is it really male-driven changes or female-driven changes, and start to understand kind of the impact of natural and sexual selection in driving the evolution of sexual dichromatism. And I am not the first one to do this. Actually, Kevin Burns, my master's advisor, did ask this very question for his PhD back in the day, and used different tanager genera and categorized them as being colorful, monochromatic, meaning males and females are the same, colorful, dichromatic, meaning that they're different, males being colorful, but dichromatic, females being colorful, dichromatic. You see, he categorized them in all these different categories, or being polymorphic, and had them on the tree. And what he found was, in fact, it was more often changes in female plumage driving the evolution of sexual dichromatism in tanagers than male plumage. This was really one of the first studies that were highlighting the importance of studying female plumage. So that was really exciting. And so why am I still asking this question? Well, there are a few things that we had to address to make sure that these results still hold true. For one, you can see that he used the tanager genera. I just showed you that now we have a molecular phylogeny at the species level. And in fact, I think it was no more, any genus of birds any genus of tanager that had more than 6 species was in fact not monophyletic, meaning that all of the species in that genus were not each other's closest relatives. So there's been quite a lot of shuffling of genera since this time. And it's really actually, plumage changes so quickly that it's important to understand how it's changing from species to species. Also, he categorized birds based on the human visual system. And why is that a problem? Well, to address that, we really need to take another step back and ask a much more basic question, which is, what is color? We think of color as being, say, reds, greens, and blues, but actually color is all in the eye of the beholder, so to speak. So simply, it's how our eyes are seeing particular wavelengths of light on the electromagnetic spectrum. As humans, we can see from about 400 nanometers to 700 nanometers. But of course, there are many more wavelengths on the electromagnetic spectrum that are important, that one can consider, ranging all the way from very, very short wavelength cosmic rays all the way to, broadcast band and radio and microwaves, for example. And so as humans, as I said, we see from about 400, 700 nanometers. We have 3 different types of cones in our eyes that are perceiving these wavelengths. And based on how these different cones are stimulated is going to determine what color our brain sees. So what our brain calls that particular color. However, birds actually have a very different visual system. So they don't have 3 cones, they actually have 4 cones. And that 4th cone is actually in the ultraviolet part of the spectrum. So they're seeing down to about 300 nanometers. So they're seeing colors that we can't even perceive. You notice that these curves are much more evenly spread out and that they're much more separated. And so there are some other aspects of the bird visual system. I don't have time to get into them, but birds can actually differentiate between much more similar colors better than we can as well. So birds simply can see color better than we can. Now let's think about this in terms of a color space, because that's how I'm going to be talking about color quite a lot. So on the left here, we have a human visual color space. And as I said, we have 3 different cones. You could think of these 3 different cones on the 3 different vertices of that color space. And based on, how stimulated each cone is by some particular light that's coming into the eye, we could think of a point being dropped in this different color space, and that's going to determine what that color is. So we could think of as human color spaces existing on a 2-dimensional plane. Birds, on the other hand, because they have 4 different types of cones, actually we can think of their color spaces existing in 3 dimensions. So not only do they have kind of this vertical, these horizontal axes, but they also have this ultraviolet axis. So there's simply a whole nother dimension of color that birds are seeing. So for example, in this painted bunch, and people ask me all the time, Allison, could you make bird vision goggles? That would be really great. I would love to see how birds see. And I too would love to see how a bird sees. But unfortunately, not only do our eyes not actually perceive ultraviolet color, sure, I could lay over where UV color is in a picture, for example, but our brains simply can't process color in this way. The closest thing I can do is actually what happens if we take out one of our dimensions of color. You can see if we take away red, a painted bunting looks very different. And so you can imagine to a bird, a color, a bird to another bird is going to look very different to how it looks to us. So, okay, so now we've told you that birds see very differently than we do. And so we need to actually study color from the bird's visual perspective compared to ours, because we weren't driving the evolution of bird color. And so how do we do that? Well, we quantify color using a device called a reflectance spectrophotometer. And so this is a device that you can use for either museum specimens or live specimens. Basically, I'll have another picture on the next slide. So here's our spectrophotometer setup. This little box here is a light source. This little box here is a speck. Then we have this kind of blue probe connecting the two. And so the light source will emit light. We use here what's called a pulsed xenon light source, so it emits light across the important wavelengths. That light is going to travel down the probe. The probe, you're going to have it next to a bird, just like this. So that light is then going to reflect back off into the spectrophotometer, which is going to feed into my computer. And so that's going to give me a reading of how much light is being reflected at each different wavelength. And I'm only considering 300 to 700 nanometers because that's what birds can see. And so for my master's, I traveled around. I wish I could have studied birds live. Unfortunately, I didn't have, however many years it would take to find all of the tanagers throughout Central and South America, as fun as that would be. But I did a museum tour instead, traveled to many different museums and collected about over 90,000 what are reflectance spectra. So of these measurements, which came from about 30,000 plumage patches. We measure the different plumage patches 3 times, so we make sure that we're not having any sorts of errors, from about 2,800 individuals from 361 species of the tanagers. So we have a very, very cool data set. Here's what a reflectance spectrum looks like when we plot it out. So here's percent reflectance, so that's like how much light is being reflected. Wavelength is on the X, and so the higher this is, the more reflectance. And so here you can see a very cool patch. This is the ear patch of a glistening green tanager. And to us, this patch looks pretty gray, but you can actually see here's where the UV cutoff is. You can see that actually almost all the reflectance is in the UV. So in fact, these glistening green tanagers, it's not that they have these kind of dull gray colored patches on their ears and on their shoulders, as well as on their cheek. They actually have ultraviolet colored patches that we simply aren't detecting correctly. So the next question we asked was, how important is this for studying sexual dichromatism? So do birds see males and females differently than we do? We think of tanagers, if you look through a field guide, they're about 55% dichromatic, so males and females are different, about 45% monochromatic. What happens when we actually take these measurements and pass them through a model of how birds are seeing vision? Well, in fact, they're about 94% dichromatic. So there's a lot of variation between males and females that we simply are not detecting. And so that means that plumage really has to be quantified from the avian visual perspective. But how do I say something's colorful or not colorful? That's a somewhat subjective term. Well, here, for example, is a paradise tanager, a bird that I think of as being pretty colorful. Perhaps you do as well. If I look at the reflectance spectra from its different patches, you can see what those look like. And what if we plot them in with the avian tetrahedral color space, that color space I showed you before? You can see there's actually a number of different measurements we can make. We can look at the volume, how much space is being occupied. We can look at the distance between or among the points, which you could think of as like how contrasting plumage is. We can look at the difference in hue of these points. And altogether, we actually came up with a metric of plumage colorfulness using these tetrahedral color space. So paradise tanager is a bird that would have a very high measure of plumage colorfulness on this metric that we came up with using principal components analysis for those of you who are statisticians. An example of a bird that scored very low is the small ground finch, one of Darwin's finches, which has pretty much just black plumage, not very exciting looking. You can see all of its points fall into one particular place on the tetrahedral color space. So very small color volume, very small distance among the points. So these are kind of like two ends of the spectrum. Now, just for fun, here's what it looks like when you plot all of the color patches of the tanagers, into a plot. This is just showing the X and the Y coordinates. You can see that there's a lot of diversity that this clade is occupying in terms of color. Now, okay, back to this question. Remember, our whole point is we want to compare male and female plumage coloration so we can try and understand the impacts of natural and sexual selection. Well, here we have that scale. So this scale I told you about, ranging from low colorfulness to high colorfulness. We have that for males and females. If males and females had the same amount of color, we would expect the points to fall like this. So we would expect them to be the same in both sexes. If males are colorful and females are not, we expect points to basically be in this lower right-hand area. So kind of pretty high on this male scale and pretty low on the female scale. Alternatively, maybe females are colorful and males are cryptically colored, and in which case we would expect the points to be up there. What's our actual data look like? How do male and female plumage compare? Well, they are actually pretty significantly correlated. But there are some points that are falling off of this one line. And those are cases where males are more colorful than females. So, this pretty much matches what we think of in birds, which is that oftentimes males are more colorful than females, but there are many, many species in tanagers where they're equally as colorful. So what do we think about how this plumage is evolving? Well, remember I told you we had the family tree of tanagers before. Here's a cartoon family tree with just a few different colorful cartoon birds. It's what we can do when we actually know the evolutionary relationships is that we can try and infer what things are back like in time. So we can do what's called an ancestral state reconstruction, where based on the current plumage colors, or any trait for that matter, we can infer based on evolutionary relationships what we think the ancestor looked like. And then what we could do was actually understand, how big of a jump was it from that ancestral state to the current state. And we can do that for each branch on the tree. So for example, we'd measure from this state to this state, and from this to this, and this to this, and this to this. And the greater the distance, the greater we can think of the evolutionary rate is. And so in fact, by doing this for males and then doing this separately for females, we can say, is male plumage evolving faster or making bigger jumps than female plumage, or vice versa? So what does it look like when we compare here? Well, you can see that, again, there are a lot of similarities between male and female plumage distance, but there are more, they actually are significantly different, and there are bigger gaps, bigger jumps in male plumage than female plumage. So this is a little bit different than what Kevin was finding back for his PhD dissertation when studying genera, suggesting that in fact actually males may be changing more than females. We're going to do one more test, which is then we're going to actually compare sexual dichromatism. So this is the difference between the sexes, to being colorful or being more cryptically colored. And we're going to do this separately for males and females. What do we find? Well, we found that there is a significant correlation for males, where species that are more sexually dichromatic tend to have more colorful males. Species that are less sexually dichromatic tend to have less colorful males. But we did not find that same relationship for females. And so, when taking all of these data together, we find an answer to our first question, which is that male plumage change is more strongly correlated with the evolution of sexual dichromatism, suggesting that perhaps sexual selection is maybe a greater driver than natural selection of plumage color. But, of course, males are still under natural selection as well. But so, we're just talking a lot about kind of the whole plumage of birds. What about different specific patches? So, for example, what if we divide a bird up into a few different patches that we could measure on all of these different species, say the crown, the throat, the breast, the belly, the back, the primaries, the rump, the dorsal tail. Do we see any difference between males and females in, for example, patches maybe that are more important for signaling? Think of these kind of ventral patches that are very important for signaling, that birds use for signaling are kind of these ones. You could think of, some species certainly use a rump for signaling, but maybe it's less common for all species. Whereas a patch like the primaries are under much stronger natural selection because that's what the bird uses to fly. So it simply doesn't have as much agency for the primaries and also probably the dorsal tail to have changes. So what we did there was we actually we took measures of color for each of these patches, hue, chroma, and brilliance, for those of you who care about such things. And we fit a model comparing those measurements to sexual dichromatism, and we compared the fit of the model for males and females. And we asked for each of these patches, was a male model a better fit or a female model a better fit? And here's what we found. We found that actually, a male model was a better fit for all of these ventral patches, for throat, for breast, for belly, and crown. We couldn't distinguish between males and females for primaries or rump or chrism or back, and a female model was a better fit for the dorsal tail. So this was very interesting. I don't exactly know why females would be a better fit for the dorsal tail. Perhaps there's just more female variation there, I'm not sure. But the fact that males are a much stronger, better fit than females for all of these patches that are important of signaling actually does suggest that sexual selection is a really important driver for changes in male and female plumage. And so in answer to our second question, we find that changes in male plumage coloration are correlated with the evolution of sexual dichromatism in signaling regions. So this is really exciting. This is showing that not only are we finding something different than was found previously by using a species level phylogeny and also looking at evolution from the bird's visual perspective, but we can actually distinguish on a bird, which patches are more correlated and which ones are not. And so now we're going to ask another question, which is, do species with more complex plumage have faster rates of speciation? What do we mean by that? Let's talk about speciation. Well, speciation, we could think of as, how quickly are species diversifying? So how quickly are species being created? Here we have two different trees. I also want to mention that this work was led by Rosalind Price-Waldman, another master student of Kevin's. So we have two evolutionary trees that actually have the same amount of time because we measure time on the x-axis. So this length is the same as this length, but this tree has many more species than this tree. And so, we wonder, why does this tree have many more species? What was it that allowed species to diversify so quickly in comparison to this tree, which had, theoretically the same amount of time, but where speciation hasn't occurred as fast? And does that relate to plumage color? So that's one hypothesis. So one hypothesis that we had was that species with more colorful plumage have a faster speciation rate than species with more cryptically colored plumage. And that's because you might think of colorful plumage as being important in sexual selection, which might increase the rate of speciation because you have, species. This color might be related to how species are able to differentiate amongst each other, which could feed into faster speciation rates. And so again, we made use of this phylogeny of tanagers. You can see here, basically in this circle type phylogeny, time 0 is in the center, and then present day is on the edges. So we do have quite a lot of variation in species, how species are accumulating through time in tanagers. But was that related to plumage color? Well, in fact, it was not related to plumage color. So it doesn't matter how fast or how slow speciation is happening, you don't tend to have either more colorful birds or more cryptically colored birds. So at first, when we got this result, you were a little disappointed. It's always disappointing when you have null results, you don't have positive results. But then we thought about this more, and we thought about, what if it's not actually the colors of the birds, but how fast color itself is evolving? And so we made another hypothesis, which is that perhaps birds where there's more speciation is happening are also diversifying their color faster. So perhaps color is evolving more quickly when species are evolving, as opposed to this example where you see species would be evolving slowly and color is also evolving slowly. Here you can see on this very fancy tree, this is the tanager tree again. Each branch is colored by how fast speciation is happening. And so you can see there's a lot of variation in either slowly diversifying species or fast diversifying species. Red is fast and kind of cooler colors are slower. And so what happened when we compared that to rates of color evolution? Well, we found that for both males and females, there is a correlation between speciation rate and plumage color evolution rate. So the faster speciation is happening, faster color evolution is happening as well. This makes sense because you might imagine that as speciation occurs, if in fact this signaling trait, which is plumage coloration, is really important, you might have that trait diverging between similar species more quickly. And so it doesn't matter if you're diverging into colorful or drab, it just matters that there's differences amongst these species possibly for species identification purposes, for example. And so this was really exciting. So we found that species, when we asked this question, we found that in both sexes, the rate of plumage evolution is correlated with rate of speciation. Now I'm going to take just a super brief break from tanagers. We just had a paper that came out last week, so hot off the presses. Diego Beltran is a student at the Universidad de Antioquia, at his advisor Juan Parra, and we worked together on evolution of color in hummingbirds. And so here's that color space. Here's male hummingbird color space. Here's female hummingbird color space. It's probably no surprise, male hummingbirds are more colorful than female hummingbirds. This is like the volume of space. You can see it's larger for males than for females. And we asked the same question about speciation rates and color evolution rates in hummingbirds. And we found that there were many significant correlations across the bird's body. So here we were looking at each patch separately. And I know there's a lot of information here, but the thing I want to tell you is that for almost every patch, there were significant correlations for both males and for females. One of the exceptions is actually the gorget, where we only have male correlations, but not for females. So this is perhaps accentuating the fact that the gorget, a signaling patch, may be more important in driving speciation rates compared to other types of patches, which maybe are equally under selection in both males and females. So, sorry, back to tanagers. Now we're going to go into how do plumage coloration mechanisms evolve differently in males and females. And so when I describe, the type of research that I do in terms of plumage color, I like to talk about how and why birds are the colors that they are. I've talked a lot about the why, but now I'm going to talk about the how. So Ryan Terrell and I, we just are finishing up a review paper on feather functions and feather evolution. This is a figure from that paper. And in it, we talk about how bird colors are produced by what we can think of as hierarchical modules. What do we mean by that? Well, it means that feathers are basically made up of these different components that contain each other. So if we think about, the entire color and pattern of a bird, it's made up of, how feathers are arranged in the body. Are they in tracks, for example? Are feathers on the head, or are they on the rump? Each of these feathers placements is made up by an entire feather itself, which you can think as the macrostructure of the feather. Feathers have different shapes. If you've ever looked at a primary versus what's called a contour or body feather, you know that they're very different. But then we can keep zooming in. So of course, feathers are made up of a central rachis with feather barbs coming off of them and feather barbules coming off of that, just like a tree has branches and a branch has leaves. And if we zoom in at these feather barbs and these feather barbules, here they look a little bit like this. We call this the feather microstructure. I'm going to talk a lot about microstructure in a moment, but we can keep zooming in. So then we can look at, how the different molecules are arranged within a feather barb or a feather barbule. That's what we call the feather nanostructure. So when we talk about structural color, for example, we're actually often talking about it at the nanostructural level, because this is how light is refracted off of these different molecules, produces things like iridescent color or blue color in birds. But then, of course, there's the molecular composition of these feather barbs and these feather barbules we could think of as the pigments, as well as keratin, which makes up most of feathers. So it's all of these kind of modular things that add up to these color patterns that we've been talking about. So we've really been focusing a lot on color and pattern. But to really understand, how feathers are evolving, we need to start diving into what's making up these feathers. So we need to start going into some of these kind of more specific modules. So I'm going to talk a lot about microstructure now. And so this all started for me when, I've always been really interested in kind of structural versus pigmentary color. But the idea of microstructure really started for me in my PhD when a fellow graduate student, Cody McCoy, she was a couple years younger than me, but she was presenting some of her undergrad work at a talk, and she was talking about this new type of color that was recently just being described by her and her advisor, Rick Crum, called super black color. And I just thought this was like the coolest thing ever. So what does this mean? Well, here we've got a typical black feather. You've got its barbules are all in a plane, so they're all pretty flat. Here we've got a super black feather of a bird of paradise. You can see not only are its barbules, are they angled, but they actually have all of these little extra kind of bits on them sticking up, which is going to collect more light. And when you're looking at feathers under a scanning electron microscope, you actually do what's called sputter coating them, which means you coat them with a precious metal like gold, for example, or platinum. And when you sputter coat a regular black feather, this black color is produced by melanin pigments. And so when you coat it by metal, that covers up these pigments and you can't see them anymore. So it looks the same color as the metal. However, when you sputter coat a super black feather, even though you're covering up this the melanin pigments, it still looks black. And that's because it's actually the shape, the position of this feather microstructure. So the position of the feather barbs and barbules that are absorbing more light than pigments can alone. And so this is the type of thing I thought that was super cool. And immediately I thought of tanagers. So it's like, oh my gosh, tanagers have to have a ton of microstructural variation. If you look at tanagers, you can see some of their feathers look shiny, some of their feathers look velvety. And in particular, I came up to Cody after her talk and I was like, Cody, there's this tanager called Ramphochelys carbo. This is the silverbeak tanager, which I had to throw out of all of my tanager color analyses, because even though I could clearly see these birds look red to my eye, they look maroon. the reflectance spectrophotometer was reading very little reflectance, so little that it didn't pass the threshold of enough to be included in my analysis. So this had always been a conundrum for me. I'd always wondered why that was. So I was like, Cody, could these birds have super black plumage structure plus carotenoid pigments? And Cody also luckily thought that was a really interesting question. And so we actually embarked on what ended up being a several-year project to look at the feather evolution of feather microstructure and pigments in these Ramphichelys tanagers. There are 10 different species in the genus Ramphichelys, and we ended up expanding beyond just Ramphichelys carbo to the other species. Here's some examples of males and females. And you can see in this group, they have a kind of a motif of being overall males will have these bright red or yellow patches, potentially also kind of these maroon patches. They've got these really bright beaks. Females will have oftentimes kind of similar motifs of color, but be much more brown, so be much more cryptically colored. And so what we wanted to know was, are the difference in the carotenoid pigments, or is it in the feather microstructure? And why do we care about carotenoid pigments? Well, carotenoid pigments are often used as honest indicators of a bird's quality for many different species. So think about our house finches, which you heard me talk a lot about last time, and which you'll hear something about next time. Well, the reddest male house finch is often sought after by the female as being kind of the best mate, whereas house finches range from yellow to red. Well, this occurs not just in house finches, but in other carotenoid colored birds as well, because carotenoids are a good indicator of a bird's metabolic state. Birds have to metabolize carotenoids that they have to eat. They have to get them from their diet to turn them from kind of your yellow dietary carotenoids to fancy red metabolized carotenoids. And so it's expensive. So you could think of as the more vivid your carotenoid patches are, kind of the more attractive you're going to be. And so that's why we were wondering, how different are the carotenoids between these males and females? And let's also look at the feather microstructure. So again, just a reminder, here's our bird feather. We've got, the feather barbs and the barbules coming off of them. Well, we analyzed the carotenoids with a technique called liquid chromatography mass spectroscopy. So we just analyze them on a very specific chemical level. And we use scanning electron microscopy to look at the structure of a feather. So first, the carotenoid results. Well, first of all, there was a lot more variation in carotenoid compounds than were previously known, but really interesting. So here we've got the phylogeny, the family tree of the Ramphichelys, the males on the top, the females on the bottom. Each of these squares indicates a different type of carotenoids. The darker the square, the more of that carotenoid in that species. The colors are what we call carotenoid families. And for those of you who are chemists, the different kind of boxes of the different colors are different isomers of that different carotenoid. The real key takeaway here is when you kind of scan the pattern in males and scan the pattern in females, you notice that they look pretty similar, right? So there's not a lot of obvious differences between the males and the females. Interestingly, we have the males in some of these species in what we call the rumped tanagers, things like the scarlet rumped and the lemon rumped tanager, for example, have evolved to have are not only using metabolized carotenoids, but adding dietary carotenoids to the mix, which is interesting because if dietary carotenoids are the cheaper type, why would they evolve? Why would they add them? It's unclear. So, but let's actually analyze males versus females in a more quantitative way. And when we look at the correlation, we find that there's a very strong correlation between how much of each different type of carotenoid are in males and how much are in females. And so we were pretty surprised by that. Because carotenoids are so important in kind of sexual signals, we expected males to have more types of carotenoids and more metabolized carotenoids than females. So what about feather structure? Well, first of all, we confirmed that male Ramphichelys tanagers have that super black feather structure, whereas females do not. So female feathers look like a typical feather. Here's the barb, here's the barbules, whereas males, they don't have the super fancy little like flanges that the birds of paradise do. Birds of paradise, as with many things, are kind of a whole nother class of feathers altogether, but they have much more dense feather barbules, and these barbules are at about a 40-degree angle. So they are capturing more light than expected. But another thing we found that was actually really cool and was unexpected is that there's a lot more variation in the overall shapes of feather barbs and feather barbules in all of the reds of the Ramphichelys males than we previously expected or we previously knew. So here's our friend, the silver tanager Ramphichelys carbo, who does indeed show a super black-like feather structure with carotenoid pigments. And so the hunch that I had, which started this whole study, was correct. But also in these really, really bright, if you ever, I wish I could show you one in the collection, because the red rumps of these tanagers are just so vivid. They're just so brightly colored. But we showed that, in fact, the barbs are much thicker, so they're much more expanded on these feathers, and the barbules are angled and they're reduced. In a few, we have these oblong barbs with angled barbules. And here, if we plot kind of barb and barbule space, here we have all of the males, so just showing that there's a lot more variation in males, whereas females all kind of cluster together. So what do we have? What do we do next? Well, we wanted to show, we did some simulations showing that this is showing that barb angle, in fact, does decrease reflectance. So this is kind of just proof of concept that that angle matters. And this is showing that basically that bigger barb matters too. So there's more light coming off of that. And so taken together with the carotenoids, this is really interesting because it's showing that males and females have the same carotenoids, but males have a lot more diversity in feather microstructure. And that diversity is to emphasize the reds. And so we just, in our paper, which was just published actually about a month ago, we hypothesized that males are actually have evolved the ability to kind of cheat this test of feather carotenoids to the females. So they're cheating by making themselves, they're enhancing their redness by, first of all, putting that bright color right next to that really, really dark black to make it stand out more, and also by changing the microstructure instead of adding more carotenoids. And where are we going from there? Well, there's a lot more species of tanagers to look at. We just looked at, Ramphichelys tanagers. So I'm excited to really delve into this feather microstructure, which I feel like is a very kind of underappreciated aspect of feathers. One thing is there any function to this variation? Does it change the ability for a bird to defend against parasites? Does it change the ability of a bird to thermoregulate? We don't really know, but I'm excited to kind of explore this aspect of tanager diversity and other birds probably as well, and then try and think about how they add up to this whole plumage color pattern. I'm collaborating with Rosalind again. She's now in her PhD at Princeton with Cassie Stoddard. So we're collaborating on a project looking at structural color and how feather microstructure is varied there. Jackie Dahl is an Oxy undergraduate student who took these beautiful photos and has expanded within Ramphichelys, expanded out to the other Tachyphonenae members. So we can look at how feather structure is evolving. You can see this iridescent colors is the blue-black grassclip. Iridescent colors actually in feather barbules, whereas other types of colors in feather barbs. And Cody and I are continuing our collaboration to look at carotenoid colors. So a lot of exciting research. You'll have to have me back in a couple of years and I can update you with what results we're finding and understanding, how and why color is the color that it is. So with that, I am happy to take any questions. And I'm going to stop sharing.
Yeah, that was wonderful. Thank you very, very much. And
my pleasure. I see more colorful talk than my last talk.
And let's see here. Well, people are forming their questions. So is it true that you have done a lot of traveling down to Central and South America, or is most of your research actually in the labs back in States?
Yeah, so I have traveled a little bit down there, but most of it's been in museums. Museum collections are just gold mines for studying different aspects of bird traits, and color is one of those. If we do a good job of protecting our specimens, feather color will stay true for many, many, many years. And so,
Mhm.
in our collection at NHM, we've got over half of all bird species. So there's a lot of surveying that one can do just with museum collections. Now, to get into some of these functional questions, I will be needing to actually have, field type data. So I am hoping to strengthen some collaborations I've been building and go down to, say, Colombia, for example,
Mhm.
when such things are possible again.
Yeah, a friend of mine, a long time ago, when he was working on his PhD, decided that the reason he wants a PhD is so that he can use it. So he decided to specialize or study French, so he could spend a lot of time in France. So I presumed that you were studying tanagers, so you could spend a lot of time in Central South America. But there may be other reasons.
Yeah, the first birds I ever wanted to study were actually turacos, and so
Oh,
I
really?
absolutely would want to spend time in Africa. Turacos are actually super cool because they're the only birds with a green pigment, which brings me to our first question that we have
Oh, there you go.
from Ken, which is, at what level of feather structure are blue and green color appearance created? Well, that's actually feather nanostructure plus feather pigments. So blue color in birds is created by light refracting off of this spongy layer matrix in feather barbs, and so you'll get blue light kind of refracting off, and the rest of the light is captured by melanin that's underlying it. Green is actually made with blue nanostructure feather color plus yellow pigments, and so except in turacos, which actually have a green pigment. So almost all greens are actually blue structure plus yellow pigments.
Mm-hmm. Interesting.
And yeah, I could see what common birds here in North America have super black feathers. That is an excellent question, Derry, and one that I hope to look at. We don't even know. We just learned about super black feathers even existing a couple of years ago. So I have a hunch that most of them are in Central and South America, just based on kind of glancing through the collection. But certainly it's worth looking at what common birds are here that might have super black feathers. Yeah.
Absolutely.
And is that factored in my research from Ken? I presume that's about the yellow, the green, and the blue nanostructure. And yes, so one of the things we're doing with Roslyn is we're quantifying not just feather microstructure, but also feather nanostructure at the same time. So we can look at how these different kind of levels of feather structure are contributing to color. So it's only by understanding all of it that we'll really understand how color is produced.
Mm-hmm. Mm-hmm.
Lily
Lily.
asks, are there any female tanagers that are more colorful than males in which their colors we can't see? That's a really difficult question to ask, Lily. I don't think we've ever looked at UV, how much UV in females versus UV in males. My hunch would be that there's not very many of them, and if there are, they're going to be a little bit more subtle, just because there are very few tanagers where there's obviously a more colorful female than a male. So we wouldn't really expect there to be a female with a lot more UV color, but you never know. That's actually something we could do with our data. So we should do that. Hopefully I can get back to you. I'll just look at that.
All right, thanks.
And Laura asks, to the human eye, does super black look different from normal black feathers? And it does. It looks very black. So you can look at a bird and it's like, wow, this bird is really, really black. Laura, so, the Ramphichelys, they look kind of like velvet. They almost look like little black holes almost. So, but there's probably some subtle variation. There's probably more of a continuum from like super black to normal black. So that's, yeah. Stay tuned. Stay tuned for more information on that. And Tom asked, for speciation, how have I factored out other variables such as geographic barriers that presumably contribute to the development of species? Great question, Tom. So as you imagine, speciation is a very complex process. And so, there are also other variables like dietary differences, geographic distribution, as you mentioned, Tom. And so we actually have a study with Kevin and a bunch of his graduate students where we look at a bunch of these different traits simultaneously and try and understand what might be driving the other one. So again, stay tuned. I would say for the analyses we kind of were agnostic to other variables, given just with the idea that we had enough species that, you know, these other variables probably weren't taking into account, or they probably didn't matter, should I say.
Absolutely. Whoa, thank you very, very much. And that if anyone else has any last minute questions, please pop them in before we bid adieu to Allison. And what is your burning research on right at the moment?
Oh, right at the moment, well, I'm doing a bunch of research, kind of trying to tie up some of my housefinch research that I presented last time.
Yeah.
And then digging into more of the microstructure research. So, and hopefully getting into some of the non-native species in Los Angeles research soon. So I want to sequence some genomes of some pin-tailed whitas and scaly-breasted munias, so we could try and understand what, at kind of a genetic level, do we see any commonalities amongst the many introduced species here, and what allows them to be introduced. So yeah, well, you know, I've got my lots of all sorts of things going on, but those are kind of the main things.
Fantastic. Kimball or Mark, do you have anything that you would like to add?
No, thank you very much, Allison. Excellent talk. And we're hoping to see you next time and learn more about
Absolutely.
colors and house finches and whatever else you want to tell us about.
Yep, I should be
Yeah.
around for a pretty long time, so you can keep having me back, and I'll give you research updates.
Fantastic.
Yeah, fantastic talk. Speaking of introduced birds, I got to thinking that the Pope could have gotten the bird flu from a bishop, because I hear they were introduced.
No, but that's an introduced species. Yeah, fantastic. Thank you very, very much, Allison. That was one very. I won't say it. Okay, I'll say it. It was a very colorful talk.
Colorful talk.
And very, very enjoyable. It's always a pleasure. And next week, we'll have John Feenstra here to talk about streaky little things. And let me just make sure we didn't miss anything in the chat. A lot of comments that this was a fascinating talk. Thank you very much.
Yeah, John Feenstra pointed out the black of a northern bishop is probably super black, and I've actually been kind of excited to get my hands on some northern bishops here, northern red bishops here, to like get some live super black colored birds. I bet they are.
Yep.
Very interesting.
And you know, it's really interesting also because John and I think once we're talking about how difficult it is to take, or maybe with Mark, how to take a picture of a bishop. Because maybe something is happening with that super.
I bet.
Yeah.
Probably the contrast between that bright, like red orange color and the black, cameras just don't like that.
It freaks them out. Yeah. Wow, that's interesting. Okay, well, so next week, please tune in, same bird time, same bird channel. And once again, thank you very much, Allison. And I hope to see you at least in the audience, if not on the actual panelists screen.
Absolutely.
Take care, everyone. We'll see you all next week.
Goodbye.
Bye-bye now.